imagine for a moment you're maybe a pig farmer.
Yeah.
And there's this hidden enemy, totally invisible, for weeks maybe. And it just starts wiping out your herd.
Yeah.
It's silent, it's insidious, and it's capable of crippling like an entire national industry.
Mm-hmm.
That's classical swine fever. You might have heard it called hog cholera historically.
Mm-hmm.
And today, well, we're taking a deep dive into why this unseen threat really demands global vigilance.
It's not just about the pigs themselves, though. I mean, it is, but it's also this highly contagious viral disease that poses a constant, pretty profound economic threat to the global pork industry.
Right. And you've given us this huge stack of comprehensive reports, detailed research. It covers everything, really, from the virus's biology, its intricate details, to its massive global impact and the strategies we actually use to fight it.
So our mission today really is to cut through all that information, pull out the most critical insights, and give you, our listeners, a shortcut to understanding this really formidable foe.
We're going to explore its, well, frankly, surprising history, the really insidious ways it spreads, the complex science behind its varied forms.
Yeah, it's not simple.
Not at all. And the massive economic ripple effects it can cause. And here's where it gets, I think, really interesting. We'll see how the battle against classical swine fever, or CSF, reveals this fundamental difference in how we approach animal disease control globally.
Yeah, that comparison is key. But before we dig in too deep, there's one crucial fact we need to get straight right up front.
Okay.
Classical swine fever is not zoonotic.
Right. Important clarification.
Absolutely. The virus doesn't affect humans. There's zero public health risk from consuming or handling pork from affected animals. Its impact is entirely confined to the health and welfare of pigs and, well, by extension, the economic stability of the whole pork industry.
Okay, good. So let's turn to that devastating history and its global footprint. This disease, it was first recognized way back mid-19th century in the U.S.
Yeah, hog cholera then.
Right. And it quickly spread worldwide, becoming one of the most feared pathogens for pig producers everywhere.
Mm-hmm. And while some regions, North America, Australia, a lot of Western Europe, they've successfully eliminated it through these massive coordinated campaigns.
Hard work.
A lot of hard work. But it remains a persistent challenge. It's endemic or pops up sporadically in significant parts of Asia, Central and South America, the Caribbean, parts of Africa too. So it's this constant threat of reintroduction to those clean zones.
What makes it so challenging, though? You mentioned its nature.
Well, yeah, it's highly variable. The severity of CSF, it's heavily influenced by the viral strain's virulence, how strong it is.
Right.
But also the pig's age, its nutritional status, the herd's overall immune health. All these factors play a role.
So it's not just one picture.
Not at all.
Mm-hmm.
This variability creates this wide spectrum of outcomes. You can get a peracute hemorrhagic fever that just wipes out nearly 100% of a herd.
Yeah.
Or you can get mild, chronic, or even subclinical infections that might go completely undetected for long periods.
And that's the scary part, right? The undetected bit.
That's it, exactly.
Yeah.
This allows for silent viral circulation, and that dramatically increases the risk of widespread dissemination before anyone even realizes there's a problem. It's a strategic nightmare for trying to eradicate it.
And we saw that nightmare play out, didn't we? In the Netherlands, 1997, 1998.
Oh, yeah. A stark example. Looking back, the analysis suggested the virus had been silently circulating for maybe 5 to 7 weeks before they even detected it initially.
5 to 7 weeks.
Yeah. And the outcome, just catastrophic. More than 11 million pigs either died or had to be culled.
11 million.
And the economic losses were estimated at a staggering $2.3 billion. It's a really powerful, chilling reminder of what this disease can do.
And the significance goes beyond just one country's disaster because CSF is, well, it's officially notifiable, isn't it?
Absolutely. It's listed by the World Organization for Animal Health, WOAH, and that designation carries a legal obligation for member countries to report any confirmed outbreak to the whole international community.
And the immediate impact of that reporting.
Instant and severe trade restrictions, live pigs, a whole range of pork products. Basically, a single confirmed case can slam export markets shut overnight.
Okay, so we've laid out the scale of the threat. Let's turn our attention to the adversary itself. What is classical swine fever virus, CSFV? Where does it fit in the viral family tree?
Right. So it's a small enveloped RNA virus. It's classified as pestivirus C, and that puts it in the Flaviviridae family.
Flaviviridae, that rings a bell.
Yeah, it should. It's the same family as some other notorious pathogens like dengue, West Nile, Zika virus.
Wow. Okay, so that's its neighborhood. Does that classification matter, practically speaking?
It really does, especially for diagnosis, because CSFV is closely related to bovine viral diarrhea virus, BVDV, in cattle, and border disease virus, BDV, in sheep. This close relationship can lead to cross-reactive antibodies,
Meaning?
meaning the tests might pick up antibodies that look like CSF, but they're actually from BVDV or BDV. So it can confuse serological tests, complicate surveillance, and potentially delay confirming a real outbreak.
Okay, so understanding the virus itself, it sounds like it has some clever tricks up its sleeve. You mentioned proteins.
Yeah, it's quite sophisticated. It starts by making this one massive master molecule, a polyprotein. Then it cleverly chops that up into smaller, specialized tools, structural proteins that form the virus particle, and non-structural ones for replication.
And are there key players among those proteins?
Definitely. Two are particularly noteworthy. One is called ERNs. It's an envelope glycoprotein, and it's unique because it has ribonuclease activity.
Like molecular scissors.
Kinda, yeah. Think of it helping the virus evade detection by degrading the host's own attempts to signal danger, and it inhibits crucial immune responses like interferon production.
Very sneaky. And the other one?
The other key one is E2, another envelope glycoprotein. This is the major surface protein, and it's also the principal immunogen.
Meaning it's what the immune system really targets.
Exactly. It's the primary target for the host's neutralizing antibody response, absolutely crucial for protective immunity, and that's why it's the protein everyone focuses on for modern subunit and marker vaccines.
Okay, here's something I found fascinating reading through the material. Despite all this, genetic variation within the virus, all known CSFV isolates belong to a single serotype. What does that actually mean for us?
It's a huge deal, practically speaking. It means antibodies against one strain generally neutralize all the others.
So one vaccine could cover everything.
Pretty much. That's the biological foundation for why single-strain vaccines, like the widely used C strain, are so successful. They offer broad protection.
But it's not completely uniform, right? You mentioned genetic diversity.
Correct. While it's one serotype, there's considerable genetic diversity within that. It's classified into 3 major genotypes, 1, 2, and 3, and various subgenotypes like 2.3, which has become the most dominant worldwide since the 90s.
And why track those genotypes?
Oh, it's indispensable for epidemiology. Genetic typing lets us trace specific outbreaks, understand how the virus is moving globally, track its spread.
Okay, so given this virus's resilience and its impact, how does it actually breach our defenses? How does it spread so well?
Well, its natural host range is pretty limited, just pigs, really. Domestic pigs, wild boar, peccaries. Infection typically starts through the mouth or nose, oral or oronasal routes. Initial replication kicks off right there in the tonsils.
Okay.
But here's the kicker. What really sets this virus apart in its ability to spread?
Yeah.
Infected pigs start shedding the virus. High concentrations. Through all bodily fluids, saliva, ionose secretions, urine, feces.
Right.
And they often start shedding 1 to 2 days before they show any visible clinical signs.
The preclinical shedding.
Exactly. That's a major, major factor in its rapid silent spread. Gets out before you even know it's there.
So what are the main ways it jumps between animals or farms?
Well, direct contact is, as you'd expect, the most common and efficient way. Sick animals tend to huddle together, which makes it easy.
Yeah.
But a really notorious pathway and a huge biosecurity risk is swill feeding.
Feeding garbage.
Yeah, feeding uncooked or inadequately heat-treated garbage that might contain contaminated pork scraps. It's a primary and highly effective way to introduce the virus. So effective, in fact, that it's banned in many countries for precisely this reason.
Makes sense. What about indirect contact?
Oh, absolutely. Contaminated objects, we call them fomites. Things like vehicles, especially transport trucks moving pigs around.
Right.
Farm equipment, feed buckets, even the boots and clothing of people moving between farms.
Yeah.
All potential carriers. This just highlights why strict biosecurity protocols are absolutely paramount.
Can it pass from mother to piglet?
It can. That's vertical or congenital transmission. The virus can cross the placental barrier in pregnant sows. This leads to infection in the womb and a whole range of devastating reproductive problems.
And this ties into something you mentioned earlier, the PI piglets.
Exactly. This is critically important. Congenital infection, especially at certain stages of gestation, can lead to the birth of persistently infected, or PI, piglets.
And they're different.
Very different. They are immunotolerant. Their immune system basically doesn't see the virus as foreign, so they don't mount an immune response, they don't produce antibodies, they'll test negative on antibody tests.
But they have the virus.
Oh, yes. They shed massive quantities of virus throughout their entire lives, often without showing any obvious signs of being sick themselves. They act as these hidden, incredibly dangerous sources of viral persistence and spread. They're the Trojan horses, essentially.
That's deeply concerning. And how long can this virus survive outside a pig in the environment?
That's another key epidemiological feature, its remarkable hardiness, especially when it's protected in protein-rich stuff, like meat or manure, and particularly at cold temperatures.
So like in meat products.
Exactly. It can stay infectious for nearly 3 months in refrigerated meat, and for several years in frozen meat products.
Years.
Yeah. Even in cured and smoked products, survival can range from, say, 17 days to over 6 months. This just underscores the huge risk posed by illegal importation of pork products and the potential for it to get back into pigs via swill feeding if those products aren't handled properly.
And we can't forget the wild populations, can we?
Absolutely not. In many parts of the world, particularly Europe, and we saw this more recently in Japan, wild boar populations serve as a self-sustaining reservoir for the virus.
So it's not just a farm problem anymore.
Right. It fundamentally changes CSF from just a domestic animal disease into this complex, persistent wildlife management issue. It creates a constant risk of spillover back into domestic farms.
Like what happened in Japan.
Precisely. The reemergence of CSF in Japan in 2018, after they'd been free for 26 years, was directly driven by the virus getting established in their wild boar population.
Okay, so what does it actually look like when pigs get sick? You said it's variable.
Incredibly variable, again, depends on the virus strain's strength, the pig's age, immune status. This variability is exactly why you can't just look at a sick pig and say, that's CSF. Field diagnosis is really, really challenging.
So let's break down the different ways it can show up. The acute form first.
Right, the acute form. This is what you often see with the really nasty, highly virulent strains hitting a herd that has no prior immunity, especially younger pigs.
What are the signs?
It's usually an abrupt onset, high fever, up to 107 Fahrenheit, 42 Celsius, profound lethargy, they just don't want to move, complete loss of appetite, they huddle together. Other really striking signs. Severe conjunctivitis, red, swollen eyes, often constipation at first, then followed by profuse yellowish diarrhea. Neurological signs aren't uncommon either: unsteady walk, hind limb paralysis, sometimes convulsions.
And that skin discoloration.
Yes, that characteristic purple-blue cyanotic discoloration of the skin. You see it especially on the ears, the snout, the belly.
And the outcome for this acute form.
Typically fatal, usually within 1 to 4 weeks. Mortality rates can approach 100%, especially in young pigs. It's devastatingly fast.
Okay, that's the acute. What about the chronic form?
The chronic form tends to develop in pigs that somehow survive an acute infection. Or maybe they were infected with a less virulent strain to begin with.
And the signs are different.
Yeah, they're often much less specific and really drawn out. You might see intermittent fever, poor appetite that comes and goes, persistent diarrhea, maybe generalized skin inflammation or dermatitis. These animals they fail to thrive. They become stunted, emaciated. Farmers often call them runts.
Do they survive?
Almost never, unfortunately. It's almost invariably fatal, but it's prolonged. It can drag on for 1 to 3 months. And crucially, these chronically infected animals are a significant source of virus shedding the whole time, continuing to spread the disease.
And then back to those congenital infections, the ones that happen in the womb.
Right. These are, from an epidemiological standpoint, incredibly important, especially with the less virulent strains. Sometimes the only clue a farmer might have is just poor reproductive performance in their sows.
Like what?
Abortions, stillbirths, mummified fetuses turning up, or piglets born very weak that don't survive.
And this is where the PI piglets come from.
Exactly. If the sow gets infected in mid to late gestation, the fetuses can become immunotolerant. They're born looking clinically normal, but they are persistently infected. They carry the virus, they shed huge amounts of it continuously, but crucially, they don't produce antibodies. They're that silent reservoir, incredibly difficult to detect without specific virus tests.
So if an animal dies, what clues might you find during a post-mortem exam, necropsy?
In acute cases, the most striking finding is usually hemorrhages. Widespread pinpoint ones, petechia, and larger blotchy ones, ecchymosis.
Where do you see those most clearly?
They're often particularly obvious on the kidneys. They can give the kidneys this characteristic turkey egg appearance because of all the little spots. You also see them on the lining of the urinary bladder, the larynx, the epiglottis.
What about other organs?
Lymph nodes throughout the body are usually severely swollen and hemorrhagic. If you cut them open, they often look dark red or kind of marbled. The tonsils are often inflamed and necrotic, dead tissue. And the spleen can have distinctive splenic infarcts.
Infarcts?
Yeah, these are wedge-shaped areas of dead tissue, usually dark red to black right along the edge of the spleen, caused by blocked blood supply. Historically, these were considered almost pathognomonic-like, a sure sign of CSF. But honestly, we see them less frequently with the more modern, moderately virulent strains that are common now.
And under the microscope?
Microscopically, you'd see a depletion of lymphocytes, those key immune cells, and characteristic inflammation in the brain and its coverings, a non-suppurative meningoencephalomyelitis, with immune cells cuffing around the blood vessels. It's a distinct pattern showing how the virus hits the nervous system.
And for chronic cases, what are the postmortem signs?
They can be harder to spot clearly because often they're complicated by secondary bacterial infections that muddy the picture.
But is there anything characteristic?
Yes. The most characteristic finding, if you see them, are button ulcers.
Button ulcers?
Yeah, they're these distinct, circular, raised, necrotic lesions. They look like they have concentric layers of this yellowish, fibrinonecrotic debris. You find them most commonly in the large intestine.
And for the congenital cases, the piglet's infected before birth.
You might see things like an underdeveloped cerebellum part of the brain, which can cause neurological problems, thymic atrophy, the thymus gland being shrunken, and sometimes various deformities of the head or limbs.
So given all this variability, the non-specific signs, you really can't diagnose it just by looking.
Absolutely not. A definitive diagnosis never relies solely on observation in the field. You need unambiguous laboratory confirmation. That's an absolute requirement before any official control measures like culling or trade restrictions can be put in place.
Which brings us to sampling. If you suspect it, getting the right samples to the lab is critical, right?
Totals. For detecting active infection in live animals, you want whole blood, preferably collected in EDTA tubes, especially from pigs showing fever. Tonsil swabs are also very useful. And for post-mortem tissues, tonsils are key, along with spleen, lymph nodes, maybe kidney, and a section of the ileum, part of the small intestine.
Okay, so what happens in the lab? How do they confirm it?
For detecting the virus itself, confirming active infection, the modern standard is molecular testing. RT-PCR, and particularly quantitative RT-PCR or RT-qPCR.
Why those?
They're incredibly rapid and highly sensitive for detecting the virus's RNA, really crucial in the early stages of an outbreak. And importantly, these tests are specifically designed to differentiate CSFV from those related pestiviruses in cattle and sheep, like BBDV and BDV, avoiding false positives.
Is virus isolation still used?
It is. It's still considered the gold standard for actually proving the presence of live infectious virus, but it's slow and labor-intensive. Takes days, sometimes weeks, to get a result. So PCR is usually the first line for speed.
What about antibody tests? When are they used?
Antibody detection is vital for surveillance, especially for proving that a region or country is free of the disease and for monitoring vaccination campaigns, perhaps. The workhorse here is ELISA enzyme-linked immunosorbent assay.
Easy to run lots of samples.
Exactly, high-throughput screening. And there are specialized ELISAs that can detect antibodies specifically against that E2 glycoprotein we talked about earlier.
And that's important for the DIVA strategy.
Precisely. DIVA stands for differentiating infected from vaccinated animals. These E2 ELISAs are critical when marker vaccines are being used. We'll get more into that. The other key antibody test is the virus neutralization, or VN, test.
What's special about that one?
It's the most specific serological test, the international standard for trade purposes. It actually measures the functional ability of antibodies in the sample to block live CSFV from infecting cells and culture. So it definitively distinguishes a true CSF infection from those potentially confusing cross-reactive antibodies caused by BBDV or BDV.
But serology antibody testing, it has limits, right?
Definitely. It can't detect infection during the early acute phase. There's a window period, at least 2 to 3 weeks, before detectable antibodies develop. And crucially, remember those persistently infected PI piglets.
The Trojan horses?
Right. Being immunotolerant, they do not produce antibodies, so they will always test negative on serology, even though they're shedding massive amounts of virus, makes them completely invisible to antibody-based surveillance. You need virus detection methods for them.
Which brings us to what you said is arguably the most critical diagnostic challenge.
Yes. What happens when two devastating diseases look absolutely identical in the field?
African swine fever,
Ah.
ASF.
Exactly. ASF is the most critical differential diagnosis for CSF. Why? Because the two diseases are clinically and pathologically indistinguishable just by looking at the pigs or the lesions during a necropsy.
They looked this up.
In the field, yes. Both cause these acute high fever hemorrhagic syndromes with incredibly high mortality. The signs overlap: significantly high fever, lethargy, that skin cyanosis, widespread hemorrhages.
Even the turkey egg
I
kidney.
mean, the turkey egg kidneys and the hemorrhagic lymph nodes. You can see those in both diseases.
So how on earth do you tell them apart?
The key distinction, and where modern diagnostics are absolutely essential, lies in the viruses themselves. They are fundamentally different. CSFV, as we said, is a small, enveloped, single-stranded RNA virus.
Right.
ASF virus, ASFV, is completely different. It's a very large, complex, enveloped, double-stranded DNA virus.
DNA versus RNA.
Exactly. And that fundamental difference in their genetic material is what we exploit. Laboratories use specific PCR assays that target ASFV's DNA genome and separate RT-PCR assays for CSFV's RNA genomes.
So separate tests for each.
Yes. Or even better, many reference labs now use sophisticated duplex RT-qPCR assays. These can simultaneously detect and differentiate both viruses in a single reaction tube, often within a matter of hours. This technology is an absolute cornerstone of modern disease preparedness and response. Being able to tell CSF from ASF quickly is paramount.
Are there other diseases you need to rule out besides ASF?
Oh, sure. Other things can cause systemic illness, hemorrhage, or reproductive failure in pigs. You need to consider diseases like porcine reproductive and respiratory syndrome, PRRS, bacterial infections like salmonellosis or erysipelas, and maybe porcine dermatitis and nephropathy syndrome, PDNS. Again, modern labs often use multiplex PCR panels that can screen for several of these key pathogens all at once, which really helps speed up the diagnostic process.
Okay, shifting gears a bit, let's talk prevention and control. What's the first line of defense?
Farm-level biosecurity. It is absolutely the cornerstone for preventing CSFV, or really any infectious disease, from getting onto a farm in the first place.
What does that involve, practically?
It's multi-layered. Rigorous control over who and what comes onto the farm: people, vehicles, equipment, establishing clear zones like perimeter buffer areas, and lines of separation between clean and dirty areas, with strict cleaning and disinfection protocols at those points.
What about people working on the farm?
Strict personnel protocols. Things like showering in and out, wearing farm-specific clothing, and boots that don't leave the premises. Thorough vehicle disinfection is crucial, especially for trucks transporting pigs or delivering feed.
And new animals coming in.
Isolating any new animals for a good period, at least 30 days, before they join the main herd. This gives time for any hidden disease to show up. And, as we've stressed, a strict prohibition on feeding uncooked garbage or swill, plus comprehensive wildlife and pest control to keep potential carriers out.
So that's the farm level. What about nationally, if CSF does get into a disease-free country?
In countries or regions recognized as CSF-free, the primary response to an incursion is usually an aggressive stamping out strategy.
Sounds drastic.
It is, but it's often necessary to eliminate the virus quickly and regain disease-free status. It involves rapid reporting of suspected cases, immediate quarantine and movement controls around the infected farm to stop spread, humane depopulation culling of all pigs on infected premises, safe disposal of the carcasses, thorough cleaning and disinfection of the farm, and then an intensive epidemiological investigation to figure out where the virus came from and where it might have spread. Trace back, trace forward.
Is vaccination ever part of the plan in those situations? Or is it mainly for areas where the disease is already common?
It depends. In areas where CSF is endemic, always present, routine vaccination is widely used. It helps control the disease, reduce the number of sick animals and deaths, and decrease the amount of virus circulating in the pig population.
Okay.
In large or rapidly spreading outbreaks, even in normally free areas, emergency vaccination can be used to supplement stamping out. The idea is to quickly create a buffer zone of immune animals around the outbreak to slow or stop the spread.
What forms does that emergency vaccination take?
There are a few options, depending on the goal and the situation. Vaccinate to kill means animals in a certain zone are vaccinated, but then later culled as part of the stamping out. Vaccinate to slaughter means vaccinated animals might be allowed to go directly to processing under controlled conditions. Or vaccinate to live, which might be used for really valuable genetic stock that you want to save.
Now, you mentioned earlier this key difference between CSF and ASF control.
Yes, and this is absolutely critical to understand the different global situations. The single greatest difference between controlling classical swine fever and controlling African swine fever is the availability of effective vaccines for CSF and the complete lack thereof for ASF.
So having a vaccine for CSF changes everything.
It gives you strategic options. It's a game changer. For CSF, control can be a flexible strategy combining stamping out, maybe emergency vaccination, or even routine vaccination in endemic areas. The vaccine is this powerful tool to reduce the viral load in the population, protect animals, create immune buffers. It makes large-scale eradication feasible, even if difficult.
Whereas for ASF,
For ASF, without any effective vaccine currently available commercially, your control options are drastically limited. It boils down to prevention through biosecurity, trying to keep it out, and elimination through stamping out if it gets in.
no creating immune population.
No option to create immune populations with vaccination. No way to slow an outbreak using vaccines. This makes ASF control much more rigid, often more brutal, relying entirely on quarantine, strict movement bans, and widespread depopulation. The economic and social costs tend to be significantly higher and harder to manage. It really is like fighting with one hand tied behind your back compared to CSF.
And this difference extends to wildlife too, right? The wild boar problem.
Absolutely. Controlling CSF in wild boar is still a huge challenge, make no mistake. It requires this complex, long-term, interdisciplinary approach, vets, wildlife biologists, hunters, government agencies all working together.
What methods do they use?
Things like intensive surveillance testing animals that are hunted or found dead, managing hunting activities, maybe targeted culling in some areas, strict bans on feeding wild boar to stop them congregating. But the really key tool, the one that makes a huge difference for CSF and wildlife, is oral bait vaccination.
You can vaccinate wild boar with baits.
Yes, it's been done successfully in parts of Europe, developing baits containing the CSF vaccine that wild boar will eat. This provides a critical tool for reducing virus circulation and building immunity in the wild reservoir for CSF, a tool that simply doesn't exist for ASF right now.
Okay, let's dig into the vaccines themselves. What types are there for CSF?
The mainstays, the most widely used and generally most effective, are the live attenuated vaccines, or LAVs.
Attenuated meaning weakened.
Exactly. The virus is weakened so it doesn't cause significant disease, but still triggers a strong immune response. The most famous example is the C strain vaccine, also sometimes called Lapinized because it was adapted in rabbits.
How good are these LAVs?
They're highly efficacious. They induce a rapid immune response. Protective immunity often develops within just 3 to 7 days after vaccination, and it's robust and long-lasting. A single injection typically provides durable immunity, often for the life of the animal. Plus, they protect against all known viral genotypes, and as we just mentioned, they can be adapted for use in oral baits for wild boar.
Sounds pretty ideal. What's the catch?
The big catch, the major drawback, is what we call the DIVA problem.
Right, differentiating infected from vaccinated animals.
Exactly. Because these LAVs mimic natural infections so closely, vaccinated animals produce the same spectrum of antibodies as naturally infected animals. So, using standard antibody tests, you absolutely cannot tell if a positive result means the pig was infected or just vaccinated.
And that's a problem for it.
For surveillance and eradication, if you're trying to prove your country is disease-free or track down actual infection during an outbreak, you need to be able to distinguish infection from vaccination. This DIVA problem is why many CSF-free countries actually ban the routine prophylactic use of LAVs. They don't want to mask potential infections.
So were marker vaccines developed to solve this DIVA problem?
Precisely. The goal was to create a vaccine that allows you to differentiate. The most common type available commercially is a subunit vaccine. It's usually based on just one specific part of the virus, that recombinant E2 glycoprotein we talked about earlier.
Okay, so it only contains the E2 protein.
Essentially, yes. It's made using genetic engineering. And the DIVA test principle relies on this. Pigs vaccinated with only the E2 protein will develop antibodies against E2, but they won't develop antibodies against other viral proteins that aren't in the vaccine, like that ERNS protein.
Ah, okay.
So the accompanying discriminatory test, usually a special ELISA, is designed to detect antibodies against ERNS, which, remember, is absent from the E2 vaccine.
So how does the test read out?
A vaccinated animal that has not been infected will be E2 antibody positive, but ERNs antibody negative.
Got it.
A naturally infected animal, whether it was vaccinated or not, will have been exposed to the whole virus, so it will be positive for antibodies to both E2 and ERNs.
That sounds like a clever solution. Does it work well in practice?
Well, while it's conceptually elegant, there have been practical limitations. Generally, the commercially available E2 subunit vaccines are considered less efficacious than the gold standard LAVs.
How so?
They often require 2 doses to achieve solid, reliable protection, whereas LAVs usually just need 1. And the onset of immunity is typically slower with the E2 vaccines. Also, the accompanying DIVA ELISAs, the tests themselves, have sometimes faced challenges with sensitivity and specificity in large-scale field use, making interpretation on an individual animal basis a bit tricky sometimes.
So comparing them head-to-head on efficacy, safety, duration.
Okay, so LAVs, like C strain, considered the gold standard for efficacy, offer virtually complete protection against actual clinical disease and death. They also significantly reduce the amount of virus shed by vaccinated animals if they do encounter the virus. They have a very long history of safe use, even proven safe in pregnant sows and very young piglets. Onset of immunity is rapid, 3 to 7 days. Duration is long-term, at least a year, often lifelong. The big downside is the DIVA issue.
And the E2 subunit vaccines.
Extremely safe because there's no live virus involved at all, but generally less efficacious than LAVs. They might not completely prevent infection, although they usually prevent severe disease, often need that primary shot and a booster for solid immunity. Onset of immunity is slower. Duration after 2 doses is decent, can be at least 9 months or so. The big advantage is they do allow for DIVA testing.
So there's a trade-off.
Absolutely. A trade-off between maximum efficacy and lifelong immunity versus the ability to differentiate infection from vaccination for surveillance and trade. The choice often depends on the specific situation, endemic control versus emergency outbreak versus maintaining freedom.
Okay, putting this all together, what's the global picture for CSF right now? Where is it? Where isn't it?
It's really a patchwork quilt globally. You have regions that are recognized as completely disease-free. North America, Australia, New Zealand, and a large swath of Western and Central Europe. And there was a major milestone recently, Taiwan. In May 2025, WOAH officially recognized Taiwan as CSF-free. This is huge because it makes Taiwan the only country in Asia currently declared free of all 3 major trade-limiting diseases: CSF, African swine fever, and foot and mouth disease. A massive achievement for them.
Wow, that is significant. But elsewhere?
Elsewhere, CSF remains endemic, constantly present in many parts of Asia, South and Central America, and some Caribbean islands. Persistence there is often driven by those low-virulence strains that circulate quietly, maintained by factors like generally lower biosecurity standards in some farming systems, the ongoing practice of swill feeding in places, and uncontrolled movements of animals. You also get sporadic outbreaks popping up in Eastern Europe and Russia, often linked to the wild boar populations there.
And the economic consequences when it does hit? You mentioned the Netherlands example. It's just catastrophic.
Utterly catastrophic. The impact is multifaceted, goes way beyond just the cost of the dead or cold pigs. You categorize costs into direct mortality, morbidity, the cost of control measures like testing and culling, increased production costs for farmers, and indirect costs, market disruptions, loss of consumer confidence sometimes, and those devastating international trade bans.
That Netherlands case, $2.3 billion.
Yeah, and what was striking about the analyses of that outbreak was how much of the loss was actually uncompensated, borne directly by the private sector. The direct losses to farmers were estimated at $423 million, and related industries like feed suppliers, processors, transporters lost around $596 million. Those consequential losses actually exceeded the direct costs covered by the government and the EU budget. It ripples through the whole economy.
And the impact on trade.
The presence of classical swine fever is arguably the single most important limiting factor for international trade in pigs and pork products globally. It effectively creates this 2-tiered global market.
Free versus not free.
Exactly. Disease-free nations have access to high-value export markets, often commanding premium prices. Countries where CSF is endemic are largely excluded from these lucrative markets or face significant hurdles and costs.
What would an outbreak mean for a major exporter, like the U.S., for instance?
It would be economically devastating. Various models predict that an outbreak triggering an immediate ban on U.S. pork exports could cause domestic hog prices to just plummet maybe by 40–50% almost overnight,
Wow.
and a prolonged outbreak lasting months or longer. Estimates suggest it could lead to industry revenue losses exceeding $50 billion and potentially over 140,000 job losses across the pork value chain. Maintaining the disease-free status isn't just an agricultural goal, it's a cornerstone of national economic policy for exporting nations.
Looking ahead then, what are the biggest hurdles still facing global CSF control?
Several persistent challenges remain. Those wild boar reservoirs are arguably the most significant hurdle to complete eradication in many regions. The circulation of low-virulence strains makes early detection based just on clinical signs extremely difficult, allowing that silent spread. Those persistently infected PI piglets, the Trojan horses, remain a major issue, invisible to antibody surveillance.
And the human factors.
Yeah, low biosecurity farming systems, particularly in some endemic regions, provide ideal conditions for the virus to just keep circulating. And then, as we discussed, the limitations of our current vaccines, the diva problem with the highly effective LAVs, and the sometimes lower or slower efficacy of the current marker vaccines.
Is there hope on the horizon? New tools?
Definitely hope, particularly in advanced diagnostics. Things like those rapid, sensitive, and specific molecular assays duplex or multiplex RT-qPCR that can simultaneously detect and differentiate CSF from ASF, and maybe other key swine pathogens, all in a single test within hours. That's a massive leap forward in preparedness.
And better vaccines.
That's the goal. The holy grail really is a next-generation vaccine that combines the best of both worlds. The rapid, robust, single-dose efficacy we see with LAVs, but linked with a truly reliable, easy-to-use DIVA principle. Research is ongoing in several areas, looking at different vaccine technologies.
So wrapping this all up, what are the key recommendations for surveillance, preparedness, response going forward?
Well, first, enhance risk-based surveillance. Move beyond just waiting for farmers to report sick pigs, passive surveillance, to actively looking for the virus in high-risk populations, testing pigs at markets, slaughterhouses, in areas bordering infected zones, using both virus detection and antibody tests strategically.
And biosecurity.
Absolutely critical to strengthen biosecurity at all levels. Continuous education and strict enforcement of those farm-level biosecurity measures we discussed, coupled with robust national-level import controls to keep disease out, and enforcing bans on risky practices like swill feeding.
Diagnostic capacity.
Invest in rapid differential diagnostic capacity. Every country, especially those involved in international trade, needs accredited labs capable of rapidly and accurately differentiating CSF from ASF using molecular methods, ideally within hours of receiving samples. Time is critical in an outbreak.
What about the wildlife angle?
Develop and implement integrated wildlife management strategies in regions with infected wild boar. This has to be interdisciplinary, involving wildlife agencies, hunters, farmers, and veterinary authorities, integrating surveillance, hunting management, and, where feasible, oral bait vaccination.
And for countries that are currently free?
Maintain a strategic vaccine bank. For CSF-free countries, having a national stockpile of effective vaccines readily available for emergency use, should the worst happen, is critical preparedness. Even if routine vaccination is banned, you need the option for an emergency.
And internationally.
Promote international cooperation and transparency. Transparent, rapid reporting of outbreaks, sharing genetic sequence data to track virus movement, collaborative research on diagnostics and vaccines. These are all essential for effective global control and ultimately the goal of eradication in more regions.
Okay. We have certainly journeyed through the incredibly complex world of classical swine fever today, from its really devastating history and the intricate science of the virus itself, to the multi-pronged global effort, the ongoing battle to control it. You've really helped us see how its silent spread, those Trojan horse PI piglets, and its remarkable ability to survive in the environment make it such a formidable adversary.
Yeah, and the battle against CSF, especially when you contrast it, as we did, with the fight against African swine fever, it really throws into sharp relief the critical role of scientific innovation, diagnostics, vaccines,
Right.
but also just stringent biosecurity, day in, day out, and that vital international cooperation in protecting our global food systems. It's a constant, ongoing reminder of how deeply interconnected our world is and how a tiny, invisible virus can genuinely reshape economies and demand just unceasing vigilance from everyone involved.
So a final thought for you, our listeners, to mull over. What truly stands out to you about the sheer ingenuity, the scientific effort, and the level of collaboration required across borders and disciplines just to protect something as fundamental as our global food supply from threats like this?